السلام عليكم ورحمة الله وبركاته. Our lab about Enterobacteriaceae. In part 1 of this lab, I will talking about Salmonella and Shigella bacteria. While in part 2, I will talking about Klebsiella and Proteus, as well as about some routine techniques that use in the lab to diagnose Enterobacteriaceae. Okay, let's start in my lecture. First of all, I will give you a general introduction about Enterobacteriaceae. So Enterobacteriaceae are large heterogeneous group, gram-negative rod. Now, the question is, what is the benefit when I tell you these microorganisms are gram-negative? Or, what this information signal to? Does it mean there is a difference in the color of cell when staining? Is that benefit for you as pharmacologist? Okay, let me explain. Gram-negative and gram-positive mean there is a difference in the bacterial cell wall structure. And this figure shows the differences between gram-positive and gram-negative cell wall structure. So, as you show, gram-positive bacteria have a thick peptidoglycan layer and no outer lipid membrane, whilst gram-negative bacteria have a thin peptidoglycan layer and have an outer lipid membrane. As gram-positive bacteria lack an outer lipid membrane, so when correctly referring to their structure, rather than staining properties, are termed as monoderms. The outer lipid membrane possessed by gram-negative bacteria means that when referring to their physical structure, they are termed diderms. And as you know, as pharmacologists, the antibiotic act via several mechanisms. One of them is the antibiotic interferes with cell wall synthesis. So, now depending on that cell wall structure, you have to prepare or investigate antibiotics against this type of bacteria. Second characteristic feature of Enterobacteriaceae is natural habitat in intestinal tract of human and animals. Third, some enteric organisms are part of normal flora, example Escherichia coli, whereas others are regularly pathogenic for humans, such as Salmonella and Shigella. Depending on the site or the source of infection, Enterobacteriaceae are classified to 3 groups: pathogens within and outside the enteric tract, and as an example is Salmonella. And second group, pathogens within the enteric tract only, like Shigella. The third group, pathogens outside the enteric tract, such as Klebsiella and Proteus. Let's start with the first group, pathogens within and outside the enteric tract, and we will talk about Salmonella as an example of that group. Okay, let's move to Salmonella. The characteristic features of Salmonella are gram-negative rod, and we understand what is gram-negative. Second, motile with Britrichus flagella. Third, urase negative. So, the question is, what is urase? And what is the benefit of urase if it's found in this bacteria? Urase is an enzyme that catalyzes the hydrolysis of urea, forming ammonia and carbon dioxide, found in numerous bacteria, fungi, and algae. Therefore, the medium will be alkaline and balance the stomach acidity to protect the bacteria, as it happened in with Helicobacter pylori bacteria. 4. Do not ferment lactose, but produce H2S. What is H2S? The benefit, or what happened to the body when the bacteria produce H2S. H2S irritates the mucous membranes of the body and the respiratory tract, among other things. Following exposure, short-term or acute symptoms may include a headache, nausea, convulsion, and eye and skin irritation. Injury to the central nervous system can be immediate and serious after exposure. 5. Salmonella is pathogenic for human or animals when ingested. The most common cause of foodborne disease in the world. Salmonella lives in the intestine of many animals, such as cows, dogs, pigs, and bears. But Salmonella typhi only lives in humans. This slide shows the Salmonella classification. Salmonella are classified phenotypically and clinically. Phenotypically, Salmonella is classified into two groups. These are Salmonella enterica and Salmonella bongoria. Most human diseases are caused by the Salmonella belonging to Salmonella enterica. Also, clinically, Salmonella are classified to two classes. These are typhoidal and non-typhoidal group. Typhoidal Salmonella causes typhoid fever and paratyphoid fever, such as Salmonella typhi and Salmonella paratyphi A, B, and C. While non-typhoidal Salmonella causes gastroenteritis and food poisoning. Example, or other Salmonella serotype. Let's move to transmission route or epidemiology and diseases of Salmonella. Human-acquired Salmonella infection by consuming of contaminated meat or animals product. Example A. B. Via direct contact with infected animals or environments. C. Via contaminated food and water, utensils, hands of someone who handles food. The disease is caused by Salmonella. There are three main diseases caused by Salmonella. These are enterocolitis, enteric fever, typhoid fever and paratyphoid fever, and septicemia. Okay, this slide illustrates the symptoms of typhoid and enteric fever. There are many symptoms associated with typhoid fever. These are fever and constipation, bacteremia, delirium, enlarged spleen occur, rose-colored macule on the abdomen, leukopenia, anemia, and abnormal liver function. The disease begins to resolve by the third week, but severe complications like intestinal hemorrhage or perforation also can occur. In this slide, I will show you the available treatment used for treating Salmonella. So in enterocolitis, after a few days, the disease usually self-limited, causing non-bloody diarrhea, and do not require medical care, except with very young and very old. Fluid and electrolytes replacement may be acquired. Second, in enteric fever, typhoid fever, and septicemia, either ceftriaxone or ciprofloxacin. Ampicillin or ciprofloxacin should be used in chronic carrier patients. Cholecystectomy may be necessary to abolish the chronic carrier state. In this slide, I will transfer to talk about Shigella as an example on the enteric pathogen only within the intestine. First of all, I will talking about the important properties of Shigella. Shigella are broad shape, gram-negative, non-motile, non-spore forming, non-encapsulated, vacuolative and aerobes, do not ferment lactose or do so slowly, do not produce H2S, highly infectious bacteria of the Enterobacteriaceae family. Shigella are classified to 4 species or serotypes. Serotype or serogroups A, involve Shigella dysentery, which include 12 serotypes. Serogroup B involves Shigella flexneri, include 6 serotypes. Serogroup C involves Shigella boydii, which include 18 serotypes. And serogroup D involves Shigella sonai, which include 1 serotype. Note: Shigella dysentery is considered the most virulent and can produce abundant cytotoxin, known as shigotoxin. But Shigella sonai is most common isolated species worldwide. Shigella causes diseases called shigellosis, which is also called bacillary dysentery. Shigella dysentery type 1 causes the most severe disease and responsible for cases in which hemolytic uremic syndrome develops. Shigella sonai produces the mildest form of shigellosis, usually watery diarrhea. Shigella flexneri and Shigella boydii infection can be either mild or severe. So, this slide shows you the route of transmission of Shigella. Humans are the only host of Shigella. Fecal oral route is the primary means of human-to-human transmission. The 5 F's of disease transmission are food ingestion, such as water, food, drinks, flies, vomit, taps, toilet, seats, etc., fingers, and feces. So what is the symptoms that associated with shigellosis? Acute intestinal infection upon ingestion resulting in mild watery diarrhea to severe inflammatory bacillary dysentery or shigellosis, manifested by severe abdominal cramp, nausea and vomiting, fever, tense muscle, anorexia, and stool containing blood and mucus. Further complications include Reiter's syndrome, which has been associated with Shigella flexneri, severe dehydration, intestinal perforation, toxic megacolon, bacteremia, toxemia, septicemia, seizure, toxic encephalopathy with headache and alteration of consciousness, septic shock, and convulsions, very rare. And hemolytic uremic syndrome, which have been linked to shigotoxin. Potent cytotoxin produced by Shigella dysentery that can also cause other necrotoxic effects. Okay, let's move to talk about the infectious dose and the available antibiotic to treat shigellosis. Infectious doses are very little, as few as 10 to 200 cells can cause disease, depending on the age and condition of the host. The treatment of shigellosis is fluid and electrolyte replacement. In most cases, no antibiotics are indicated. In severe cases, a fluoroquinolone, example, ciprofloxacin, is the drug of choice. But the incidence of plasmid conveying multiple drug resistance is high enough that antibiotic sensitivity test must be performed. Trimethoprim, sulfamethoxazole is an alternative choice. Antiperistaltic drugs are contraindicated in shigellosis because they prolong the fever, diarrhea, and excretion of the organism.